Now our topic is about osmosis. So prior going into the topic, we have came through certain terminologies. Osmosis means higher concentration, lower concentration, osmosis gradient, diffusion. So with all these terminologies, it was quite confused. But now we are going to clarify all these things very clearly, and that too line by line what is happening in the osmosis. So osmosis, it is a spontaneous net movement of solvent molecules. So here the solvent means nothing but water. So water we call it as universal solvent. Whatever the thing we will dissolve in that solvent through a partially permeable membrane. So why we have opted for partially means, so some of the contents only will get moved through the partially permeable or semi-permeable membrane, not all the contents, into the region of higher solute concentration, which means hypertonic solution. So from a hypotonic solution to the hypertonic solution, the water will move in the direction that tends to equalize the solute concentration in the both sides. In order to equalize the solute concentration, the water will move from higher concentration of the water, where the water will be higher in concentration at hypotonic solution, to hypertonic solution. So osmosis provides a primary means by which the water is transported in and out of the cells. Okay. So difference in concentration between the solutions on the either side of the semi-permeable membrane, we call it as osmotic gradient. In this picture, we can able to see what is osmotic gradient clearly. So concentration gradient. So gradient means change. So the change in the concentration of substance from one area to the other area. For example, we have a beaker. So in this beaker, one side we have one concentration and the other side we have the other concentration. So it created a change or not. So this we call it as gradient. So osmosis is the movement of water across the semi-permeable membrane. At first, the concentration of solute is very high on the left side. But over the time, the water moves across the semi-permeable membrane and dilutes the particles. In this picture, this side the water concentration is more, and this side the salt concentration is more. So now the water is moving from higher concentration to lower concentration. So now the water level is rising at this side. So this is finally clear that osmosis means nothing but the movement of water from higher concentration to lower concentration. Next, in this also we can able to see the higher water concentration. This is hypotonic solution and this is hypertonic solution, where the lower concentration. So the water is moving from the higher concentration to lower concentration of water. So osmosis, it is a special kind of diffusion. So the diffusion of water across the selective permeable membrane, a barrier that allows some substance to pass through but not others. Because of that only we selected selectively permeable. The cell membrane is such a barrier. Small molecules will pass through, for example water. If the water is passing, then only we have to call it as osmosis, otherwise we have to call it as diffusion. So the larger molecules can't pass through this, for example proteins and complex carbohydrates. This is the semi-permeable membrane. So all the biological membranes are semi-permeable membrane made up of phospholipid bilayers. So this happens with simple diffusion. It is very easy that it is a type of passive transport. So there is no use of any energy, no ATP. So the water is moving from the higher concentration of water to lower concentration of water. So in this picture also, the hypotonic solution. So solution on the one side of the membrane where the solute concentration is less. On the other side, the solute concentration is high. So in this picture, it is clear that, so here we have the semi-permeable membrane, right? This side we have the concentration of water is higher, and this side we have the concentration of water is low. But this side the solute concentration is less, and here the solute concentration is more. So now the water is moving from higher concentration of the water to lower concentration of water. So from here the water is moved to this side to equalize the both sides. So now the water level is being raised here. Over time, the molecules will move across the membrane until the concentration of solutes are equal on the both sides. So this type of solution we call it as isotonic solution. Initially, this is hypertonic solution. This is hypotonic solution. So now in this case, this we call it as isotonic solution. Both the sides, the solute concentration is same. Next, cytoplasm is a solution of water and solids. Solutes are dissolved in the water. So the water moves in and outside of the cells because of the different concentration of the solutes. So different kinds of cell react differently depend on the solution they are in. So before the examples, so below the examples of RBCs, red blood cells, in different types of solutions. So what happens to the RBC? For example, if it is being placed in the hypertonic solution, now the shell will get shrinked. When the RBC is placed in the isotonic solution, there is no any change. So there will be inward and outward. When the RBC is being placed in the hypotonic solution, the water will enters inside to the cell. So these are the changes we'll see for the RBC. Next, so this we call it as turgid cell. When the water enters by osmosis and the vacuole swells and pushes against the wall, which is very rigid in nature. Next, we have the flaccid cell, which is very loosely arranged. The water lost from the cell, the vacuole shrinks and the cell loses its shapes. Osmosis in animal cells. There is a greater concentration of free water molecules outside the cell than the inside. So the water diffuses into the cell by osmosis and the cell will swells up. Coming to the plant cell, we have the cell wall and vacuole. So the cell absorbs the water by osmosis. Here, the cytoplasm and the cell membrane. But the cell wall will stop expanding anymore because it can extend to some extent only, because it will be stopped by the cell wall because of the rigidity. For osmosis, we talk about the potential water molecules have to move. So that we call it as osmotic potential. So the distilled water have the highest potential because the distilled water is lack of any salts. When the water has another substance dissolved in it, the water molecules will have less potential to move. So this osmotic potential we call it as negative osmotic potential. The osmotic potential of a cell, we know it as water potential. For animal cell, the osmotic potential of the cytoplasm. The water potential is the osmotic potential of cytoplasm. An animal cell with water potential 50 is placed in a solution. If the osmotic potential of the solution is less or negative than the water potential of the cytoplasm, the solution we call it as hypotonic. So net is endoosmosis will occur, means that is the water will move into the cell from the solution. The results will be hemolysis, means the cell will burst. So for example, when we placed in a solution, here the inside we have the less or the negative potential than the outside. When the water will enter into the cell and now the cell will burst. So the water potential of a cytoplasm equal to 50 and the osmotic potential of a solution equal to 20. If the osmotic potential of a solution is more or negative than that of the water potential of the cytoplasm, so the solution is hypertonic. So the net exosmosis will occur. So the result will be crenation, means the cell will shrivel up. So in this is the hypertonic solution. When compared to the cytoplasm, now the water will move from the cytoplasm into this surrounding solution. So now the cell will shrink. If the osmotic potential of the solution is same as the water potential of the cytoplasm, so then the solution is said to be isotonic. There will be no any net movement. So now the cell will be very stable. In animal cell, the water potential is equal to the osmotic potential of the cytoplasm. But this is different in the plant cells. The animal cells have the cell wall. The plant cells have the cell wall, which exerts an inward pressure when the cell is turgid. This is known as pressure potential. So the water potential of an animal cell is equal to the osmotic potential of the cytoplasm plus the cell wall pressure. So that is equal to water potential is equal to osmotic potential plus cell wall pressure. A plant cell with water potential 50 is being placed in a solution. If the solution is hypotonic, the net is endoosmosis will occur and the cell will become turgid. If the solution is hypertonic, net is exoosmosis, causes the plasmolysis and the cell membrane plus away from the cell wall. So the cell membrane will pull away from the cell wall, so the cell wall stays intact. If the solution is isotonic, there is no net osmosis occur, so the cell is not plasmolyzed, but there is no fully turgid pressure, either will not be there. So conclusion, osmosis is a vital process in the biological systems, as biological membranes are semi-permeable membranes. Osmosis is responsible for the ability of the plant roots to draw water from the soil from higher concentration to lower concentration. Osmosis is a vital process in both plants and animals to maintain their own life process. So I think you have cleared about what is osmosis, what is osmotic pressure, what is positive osmotic pressure and what is negative osmotic pressure, what is hypertonic, hypotonic and isotonic. So thank you very much. Please do like, share, comment and subscribe to my channel.